The story begins in 2020 at Tufts University, where researchers accomplished something that blurs the line between organism and machine. They took cells from African clawed frog embryos, specifically Xenopus laevis, and created tiny living forms called Xenobots. What makes these remarkable is that they are not genetically modified, they are not built on any artificial scaffolding, they are simply cells doing what cells do. When given the chance to organize themselves in a new context, when scientists removed precursor skin cells from these embryos and let them develop in a dish, something unexpected happened. The cells spontaneously formed small spherical structures. These little spheres became covered in tiny hair-like projections called cilia, and those cilia began to beat in coordinated patterns. This coordination allowed the Xenobots to swim through water. They could move, they could explore, and they could interact with their environment. These living bots survive for about 9 or 10 days, running on nutrients stored in the original embryonic cells. They have a remarkable ability to heal themselves when injured. Perhaps most surprising of all, they can gather loose cells from their surroundings and assemble them into new Xenobots, essentially building their own siblings. This was the foundation, this was the starting point before anyone added a nervous system, before anyone thought about neural networks or enhanced behavior. These were simply biological entities doing something unexpected. They were cells that had never existed in this form before finding their own way to be alive. The research behind neurobots was not simply about creating something new for novelty's sake. Michael Levin, the Vannevar Bush Professor of Biology at Tufts, and Hale Fadawat, a senior scientist at the Wyss Institute, had a deeper purpose driving their work. They wanted to understand something fundamental about how nervous systems organize themselves. Traditionally, scientists study neurons within existing animals. Fadawat herself had spent years examining how neurons give rise to behavior in creatures like zebrafish. But there is a limitation to this approach. When you study an animal that has evolved over millions of years, you are looking at a nervous system that has been shaped by countless generations of natural selection. Every neural connection, every pathway has been refined by evolutionary pressures to help that creature survive and reproduce. The nervous system, you observe, is the product of a long history, not a clean slate. This is where the reverse engineering approach comes in. Rather than taking apart an existing nervous system to see how it works, Levin and Fadawat wanted to build one from scratch. The question they asked was simple but profound. What happens when you give neurons a completely fresh canvas? What do they do when placed in a structure that evolution has never encountered? The Xenobot provided exactly this novel context. Here was a tiny living form assembled from frog cells, but arranged in a way that nature had never tried before. No evolutionary history guided its neural architecture. No ancient environments had shaped its connections by adding neural precursor cells to this new kind of body. The researchers could watch nervous systems organize themselves without the weight of millions of years of adaptation. This approach offers a window into the basic rules that govern neural self-organization. It reveals what neurons do innately, what patterns emerge simply because that's what neurons do, rather than what patterns exist because they help some ancient ancestor survive. Understanding these fundamental principles could eventually help scientists guide cells to build new structures in the lab, or restore damaged tissues in the human body. The creation of a neurobot came down to a carefully timed intervention. Researchers needed to implant neural precursor cells during a brief window when the spherical biobots were still forming. This was not something that could be done at any stage. The developing structures had to be at just the right point in their assembly for the implantation to work. The team started with cells harvested from early embryos of the African clawed frog. These neural precursor cells were destined to become neurons, but instead of letting them develop in isolation, the researchers carefully placed small clusters of these cells into the center of the forming biobot structures. Timing was everything. The spherical biobots were assembling themselves, and the researchers had to catch them during that narrow formation period. Once implanted, the neural precursor cells began to mature. They developed into neurons and extended branching projections outward. These projections, called axons and dendrites, spread throughout the interior of the bot and even reached toward the outer surface. What makes this approach so different from traditional neuroscience research is the context. Scientists have studied neurons in laboratory dishes for decades. They have examined small samples of neural tissue under controlled conditions, but those studies always lacked something critical. A neuron in a dish has no body. It has no way to exhibit behavior or respond to an environment. The neurobot changed that equation entirely by implanting neural cells into a developing biobot. The researchers created something new. They built a nervous system inside a living body that could actually do something. The neurons could organize themselves and potentially influence how the bot moved through its watery environment. This was the essence of the experimental technique. Take neural precursor cells from frog embryos. Wait for the brief window when biobots are forming. Implant the cells into the center of the structure, then watch as those cells mature and extend their branching networks throughout their new home. Under the microscope, something remarkable began to take shape. The implanted cells did not stay as simple precursors. They matured into fully formed neurons and began extending themselves throughout the tiny spherical bodies. Long branching projections reached outward from the cell bodies, pushing through the interior and creeping toward the outer surface. These were axons and dendrites, the classic hardware of nervous systems, sprouting in a context where no nervous system had ever existed before. The researchers could see the telltale signs of genuine neural architecture. The branches were not random. They followed patterns that looked strikingly familiar to anyone who studies developing nervous systems. It was as if the cells carried within them an ancient blueprint, one that knew how to build communication networks, even without an evolutionary history to guide them in this specific environment. But structure alone tells only part of the story. The team needed to know whether these neurons could actually talk to each other. They used calcium imaging to find out. This technique tracks the movement of calcium ions, which flood into neurons when they fire electrical signals. What the imaging revealed was activity. The neurons were not just sitting there looking like neurons, they were behaving like them, two passing signals through their newly formed networks. The researchers also identified protein markers associated with synapses. These are the contact points where neurons meet and exchange information, the tiny gaps across which chemical messengers travel. Finding these proteins meant that the cells were not just growing branches, they were building the infrastructure for communication. The pieces were in place for a functioning, if primitive neural network. The implications were profound. Raw cellular materials, given the right conditions, will organize themselves into something that looks and acts remarkably like a nervous system. When the researchers watched their neurobots swim for the first time, they saw something immediately different. The original Xenobots moved through water, using coordinated beating of their hair like cilia, and their movement followed predictable patterns. But neurobots behaved differently. They took on new shapes and displayed unique swimming behaviors that their predecessors never exhibited. The presence of neurons changed how these tiny organisms moved through their environment. This was the key question the researchers wanted to answer. Would a nervous system modify the way a biobot behaves? The answer turned out to be yes, and in ways that surprised the team. What makes this finding so interesting is that these neurons had never existed in a frog in this configuration. They had never been shaped by millions of years of evolution. There was no pre-existing blueprint for how they should function, yet they self-organized and began influencing behavior. Anyway, Michael Levin described this as fundamentally different from studying neurons in a laboratory dish. When you observe neurons in a petri dish, you see cells behaving in isolation. But neurobots have a body, they can actually do something. This allowed the researchers to observe a direct relationship between neural architecture and behavior in a living system for Hale Fadawat. This represented a form of reverse engineering. Instead of studying how existing animals produce behavior, she could watch a nervous system develop from scratch and see what behaviors emerged. The neurobots became a window into understanding the basic rules that govern how nervous systems give rise to action. The behavioral differences were measurable. The neurobots swam differently. Their movement patterns showed complexity that suggested the neurons were doing more than simply existing within the structure. They were actively participating in shaping how the organism moved. The neurobot research sits at the intersection of several rapidly advancing fields that are reshaping how we think about medicine and engineered life. Organoids research, for instance, has shown us that cells can self-organize into miniature versions of organs when given the right conditions. Scientists have grown tiny brain organoids, liver organoids, and kidney organoids in laboratories around the world. But what neurobots add to this picture is something different. They show us what happens when you introduce a nervous system into an entirely novel context, one that evolution never designed for. This matters because the same self-organization principles that guided those frog neurons to form networks could potentially guide cells to repair damaged tissues in human bodies. Consider spinal cord injuries where the nervous system struggles to regenerate. If we understood the rules that tell neurons how to organize themselves, we might be able to coax damaged tissue into rebuilding, rather than scarring. The connection extends to body-on-a-chip systems as well. These are microfluidic devices that contain living cells designed to mimic organ functions for drug testing. Adding neural components to such systems could create more realistic models of how drugs affect not just isolated organs, but the communication networks between them. A heart on a chip is useful, but a heart connected to a nervous system on a chip tells us far more about how medicines actually work in a living body. Then there is the emerging field of bioprosthetics. Traditional prosthetics are mechanical devices, but prosthetics aim to incorporate living tissue. Understanding how neurons self-organize could help engineers create prosthetic limbs that integrate more naturally with a patient's own nervous system. Imagine an artificial arm that does not just respond to nerve signals, but actually contains living neural tissue that connects seamlessly with the body. What makes all of this possible is a shift in thinking. Rather than trying to micromanage every cell's position and behavior, researchers are learning to work with the innate intelligence of cell collectives. The cells already know how to build complex structures, they just need the right context and raw materials. This approach, sometimes called synthetic morphology, represents a fundamentally different philosophy for both synthetic biology and regenerative medicine. The neurobots are small, but the questions they help answer could transform how we heal bodies and build new forms of life.